Hello friends, welcome to yet another intriguing edition of Biology Made Easy. Today I'll be dealing with the replication in prokaryotes, the first part of the elongation. I'll be breaking down the elongation portion into two different parts. In my previous video, I've already described in length about the replication in prokaryotes, the initiation part. Now let's get down to business about the elongation part. In this very video, we'll be talking about the various enzymes and their mechanistic features, their molecular details governing the elongation of the DNA replication. Now, specifically in Escherichia coli, there are 5 types of DNA polymerases: Pol1, Pol2, Pol3, Pol4 and Pol5. These are the genes responsible for encoding these specific DNA polymerases. Pol-A gene encodes Pol1, Pol-B, Pol2, Pol-C, Pol3, Din-B, Pol4 and UMU-D prime 2C for Pol5. The main functions of DNA Pol1 is in DNA repair, Pol2 is in DNA replication restart, Pol3 is the major DNA replicase. I'll just show you how. Pol4 is the translation polymerase, wherever there is translations or nick translation processes, which I'll be dealing in my later videos in length. And Pol5 also in the nick translation processes. Now, starting with the DNA Pol1. DNA Pol1 is a monomeric enzyme. Number 1, it has the 5 to 3 prime polymerase activity. You must be prudent to know that there is no polymerase which can polymerize from the 3 prime to 5 prime direction. All the polymerases have the 5 to 3 prime polymerase activity. Now DNA polymerase has one special activity known as the 5 to 3 prime exonuclease or proofreading activity. The exonuclease is also known as proofreading activity. Now it's the only polymerase which has got the 5 to 3 prime exonuclease activity. There is no polymerase in the eukaryotic system which has got the 5 to 3 prime exonuclease activity. That's why in order to remove the multiple primers in the lagging strand or the Okazaki fragments in the eukaryotic system, there is a specific enzyme known as FEN1 or flap endonuclease 1, whereas the same process is conserved or is processed by the 5 to 3 prime exonuclease activity of DNA polymerase 1 in the case of prokaryotes. Now it has also got the 3 to 5 prime exonuclease of proofreading activity, which would keep on trimming the unpaired or you can say mismatched DNA nucleotides from the 3 prime end. Now in DNA, in what is called genetic engineering, we go for the synthesis of the cleno fragment because we do not want the 5 to 3 prime exonuclease activity in our genetic engineering experiments, because the 5 to 3 prime exonuclease activity would keep on trimming the bases from the 5 end and then it would find the mismatch. So all the ATPs would be wasted. That's what we don't want. So what we do is we trim the fragment which codes for the 5 to 3 prime exonuclease, which is a 35 kilodalton fragment. So we subtract it from the 103 kilodalton which makes the complete DNA polymerase 1, and what we get is a 65 kilodalton protein which consists of the 5 to 3 prime polymerase activity and the 3 to 5 prime exonuclease activity, and that protein is known as cleno fragment. In some experiments we also use the exocleno, where we trim the 3 to 5 prime exonuclease activity also. We just use or make use of the 5 to 3 prime polymerase activity of DNA Pol1. Now after DNA Pol1, we come down to DNA polymerase 3. Now DNA polymerase 3 is of major interest because DNA Pol1 can only have the speed or can only attain the speed of 10 to 20 nucleotides per second when it comes to DNA synthesis on the new daughter strand. Whereas we know in Escherichia coli the process is about 1000 nucleotides per second in when you talk about the speed. Now it's 750 to 800 when you want to be precise nucleotide per second. Now how does the speed vary so much, and why does it not being attained by DNA polymerase 1? Number 1, DNA polymerase 1 doesn't have any kind of clamps known as the clamp subunit. It's a monomeric protein. Thereby it cannot hold on to the substrate properly while proceeding with the reaction or while proceeding with the addition of deoxyribonucleotides, right? DNTPs. So what happens is this cannot happen in the absence of beta clamp, which is present in DNA polymerase 3. So DNA polymerase 1, it's on its own, it's a monomeric protein, it cannot cause the addition of DNTPs incessantly, right? It keeps on falling off from the substrate. That's why the speed that it can attain maximally is only 10 to 20 nucleotides per second. Thus its processivity. Processivity means how fast you can process, how fast you can add the nucleotides, and how long do you not fall from the template strand. So its processivity is pretty low compared to the DNA polymerase 3. Whereas DNA polymerase 3 is a multimeric enzyme, right? It's made up of 10 different polypeptide chains, at least 10 different polypeptide chains in the prokaryotic system. Its mass is almost 950 kilodaltons. It's a huge hollow enzyme. When we talk about DNA polymerase 3, we have to talk about DNA polymerase 3 hollow enzyme, because all these subunits together, they make the functional DNA polymerase 3, which is known as the DNA poly 3 hollow enzyme. Then it has got high processivity due to the beta 2 clamp. Now understand this extremely carefully. What happens is, this is the core polymerase, the alpha, the epsilon and the theta subunits. This is known as the core polymerase. The rest, when attached to the core polymerase, is known as the complete hollow enzyme, right? So the alpha subunit is responsible for the 5 to 3 prime polymerase activity. The epsilon subunit is responsible for the 3 to 5 prime exonuclease activity, whereas the theta subunit is responsible for activating the 3 to 5 prime polymerase activity. So this is the complete setup of the core polymerase. Now this beta 2, because 2 beta subunits are there, that's why I've written beta 2. Beta 2 is the clamp, is the part of the protein, or these are the polypeptides which hold on to the template just like a clamp. It holds on perfectly and efficiently to the substrate, that is the template DNA strand, and thereby enabling the core polymerase enzyme to catalyze the SN2 nucleophilic transferase reactions in order to keep the DNTPs added, in order to cause the phosphodiester bond forming without the dissociation or without the falling off of the core polymerase from the template strand. Thereby DNA polymerase 3 can attain at least 750 nucleotide per second of the speed, and it doesn't fall off after 750. It keeps on going and it falls off after many thousands of nucleotides. Now what enables this specific beta clamp to load? What enables its loading onto the DNA strand? The answer to that question lies in the clamp loader, which is also an ATPase. Clamp loader is this part, the gamma part, the phi part, the delta dash, delta. So this is the clamp loader. Clamp loader, you can see the distribution of clamp loader is not symmetric. You can see alpha, epsilon, theta, 2 beta, 1 tau. Alpha, epsilon, theta, 2 beta and 1 tau are completely symmetrically distributed, half and half. But you can see the distribution of the clamp loader is asymmetric in nature. That's why this complete DNA polymerase 3 hollow enzyme is also known as asymmetric dimer. It is a dimer, but due to the asymmetric nature of the distribution of the clamp loader, it's known as an asymmetric dimer. Rest are symmetric except for the clamp loader. Now clamp loader, what it does is, the specific clamp loader, the theta part, it interacts with the ATP and it causes the ATP degradation, dissociation of ATP into ATP plus PI, and the energy produced, it's still a topic of huge research. The energy produced can either cause the loading or can cause the complete dissociation of the clamp after the clamp loader puts or it can load the complete clamp and the core polymerase onto the DNA. So either of the two things can be done. Either it can dissociate earlier or it can dissociate after the loading. So both the things can be done, or the energy used can be utilized by the clamp loader in order to load the entire beta clamp associated with the DNA polymerase core enzyme onto the template. So this is a thing of debate in research. Now the entire thing you can see is connected by 2 tau subunits. So tau is a symmetrical dimer here. If we talk about this specific subunit, and the 2 asymmetrical dimers are connected by the tau subunit, and thereby it is able to completely latch on, hold on to both the template strands and catalyze the formation of the DNA daughter strands via SN2 nucleophilic reactions, which will be dealt in detail in my elongation part 2 video, which I will be making next. So, and one more thing, you can see there is no 5 to 3 prime exonuclease activity in the case of DNA polymerase 3. This epsilon subunit is only responsible for 3 to 5 prime exonuclease activity. As I had told you earlier in my current video only, that there is no polymerase except for DNA polymerase 1 which possesses the 5 to 3 prime exonuclease activity. So there is none 5 to 3 prime exonuclease activity in the case of DNA polymerase 3. But it's a very processive enzyme, it's a huge multimeric enzyme 950 kilodalton, and it's an asymmetric dimer which makes use of ATPase also via its clamp loader, which causes it to become an asymmetrical dimer, right? So I think you were able to comprehend the intricate molecular details governing the elongation pattern of the replication in the Escherichia coli, which is a prokaryote. So if you have any queries, any comments, do not hesitate to put it in the comment section below. And if you do find the content relevant, then kindly hit the like button and also subscribe to my channel, Biology Made Easy. Thanks a lot. See you soon.